and welcome to my talk about the preferential formation of ATP from acetyl phosphate and ferric iron. My name is Silvana Pinna, and I'm a PhD student in the Lane Origins group at UCL in the UK, and I'm very happy to participate in this virtual AbGradCon 2021. Now, we know that metabolism is a quite complex network of life-sustaining chemical reactions, and as we can imagine, this complexity was acquired after years of evolution. So it's important to understand how we went from proto-metabolism to modern metabolism. We can envision life to have started from simple molecules such as hydrogen and CO2. For this autotrophic origin of life, we can take as example a couple of very simple modern organisms such as methanogenic archaea and acetogenic bacteria, whose metabolism you can see pictured in here. These organisms can thrive off H2 and CO2 alone, and as you can see here, it goes up here to the acetyl-CoA pathway to form acetyl-CoA. This goes to the right to carbon metabolism and to the left to energy metabolism, where they form acetyl phosphate, and this can in turn phosphorylate ADP to form ATP, which can then go on to perform all its key reactions. Often referred to as the universal energy currency, ATP is a key molecule that drives metabolism by participating in phosphorylation and condensation reactions, which build complex small components of life, such as peptides and RNA and DNA. But because of the deep conservation of functions carried out by ATP, this must have arisen early. However, like other key molecules in metabolism, such as acetyl-CoA, which we saw earlier, ATP is a complex molecule to make, so there must have been a prebiotic precursor that could carry out the same functions. Now, Kristin proposed that inorganic phosphate was introduced in metabolism via deactivation by energy-rich thioesters, which are molecules that can be made geochemically. Acetyl phosphate, which we saw earlier in the energy metabolism scheme, could have formed prebiotically this way. Now, acetyl phosphate is quite convenient for this because it's highly reactive, as it has a higher phosphorylating potential than ATP. It's universally conserved. It's the primary energy-rich intermediate with acetyl-CoA in some bacteria, and it is very proficient at phosphorylating. Now, acetyl phosphate was successfully synthesized from thioacetate and phosphate under ambient or mild prebiotic conditions in water, and it has been observed to promote phosphorylation of key molecules such as ribose to ribose 5-phosphate and adenosine to AMP. However, it cannot promote polymerization of either amino acids to form peptides, as it tends to acetylate them, nor it can polymerate nucleotides to form RNA, as the condensation reaction liberates water, and this is not favored as a leaving group in an aqueous environment. In fact, ATP liberates pyrophosphate rather than water. So the question is, can acetyl phosphate phosphorylate ADP to form ATP so then this can be selected to do its own thing? Quite serendipitously, in the 90s, Kitani et al. Found that this can happen in the presence of ferric iron. And as you can see, our repetition worked when you can see the ADP goes down and ATP signal goes up. So we decided to explore this more fully to see if this mechanism actually holds prebiotic relevance. Surprisingly, we found that ferric iron is uniquely effective at catalyzing ADP phosphorylation, or at least among the large panel of metal ions we tested, even more than magnesium and calcium, which was surprising as they are commonly associated with ATP metabolism, so we expected them to work. High-end sulfur clusters chelated by monomeric cysteine initially seemed to work as well, but it was later found out that repeating the experiments under more strictly anoxic conditions this actually reflected the release of ferric iron into the medium. Exploring the conditions under which this reaction occurs, we found that it's strongly sensitive to pH and occurs most readily under mildly acidic conditions. It's less acutely sensitive to temperature, at least between 20 and 50 degrees, and we found that 30 degrees optimized yield across 24 hours. But as these processes take place over minutes to hours, temperature really has a relatively trivial effect, as the yield between 2 to 3 hours is pretty much the same at all three temperature studies, around 10 to 15%. Surprisingly, ATP yield was greatest at high water activity, either in HPLC grade water or in suspended silica. Higher magnesium and calcium concentrations as part of a modern ocean mix collapse the ATP yields to nearly zero, and this is presumably because calcium and magnesium promote ATP hydrolysis. High pressure had very little effect on ATP synthesis, which is consistent with a variety of areas on Earth. When comparing acetyl phosphate with a panel of 8 other potentially probiotic phosphorylating agents, including a number still used by cells today, we found that ACP was the most effective. We next explored the propensity of ACP to phosphorylate other canonical nucleotide diphosphates, as well as the diphosphate with the purine precursor inosine, and we found that ACP can phosphorylate ADP but not any other nucleotide diphosphate, which demonstrates a dependence on the structure of the nucleobase. The fact that not even the other purines were phosphorylated suggests that the critical moiety is the amino group at N6. We explored the dependence of phosphorylation on the nucleobase by substituting ADP with pyrophosphate, but no triphosphate was detected by phosphorus NMR. And this suggests that the adenine ring does indeed need to interact directly with ferric ion. We also considered whether ferric ion could interact with the adenine ring but not the diphosphate tail, analyzing the phosphorylation of AMP to ADP. And as you can see here, it was successful, but the considerably low yield of ADP suggests that ferric ion interacts preferentially with the purine ring coupled with the diphosphate tail. To understand how ferric ion catalyzes the phosphorylation of ADP to ATP, we tested the effect of varying the ferric ion concentration, which showed that the maximal ATP yield was produced by 1 millimolar ferric ion, which means that the optimal ADP to ferric ion stoichiometry of the reaction was about 1:1. We confirmed what Kitani had found, that low concentrations of magnesium or calcium slightly increase the ATP yield in the presence of ferric ion. And this suggests that either divalent cation can stabilize the newly formed ATP and liberate the ferric ion so that it can continue to phosphorylate some more ADP. A kinetic study of the phosphorylation reaction gave a curve that resembles a characteristic Michaelis-Menten mechanism for an enzyme, which indicates that ferric ion does indeed act as a catalyst. And lastly, we wanted to see whether a single ferric ion was interacting directly with the single ADP and ACP, or whether larger units such as stacked ADP rings were involved. And Malditov analysis showed no difference between the ADP control and the reaction sample. There was no stacking evident. Altogether, our results suggest that the high charge density of ferric ion allows it to interact directly with the N6 amino group of the purine ring, while anchoring ACP in position for the phosphate group to interact with the diphosphate tail of ADP. The interaction of ferric ion with the diene ion is key because at the optimal pH of 5.5, the first two hydroxyl groups of ADP are deprotonated, while the external OH group remains protonated and is therefore not available for nucleophilic attack. So the interaction of the ferric ion with the two deprotonated OH groups has the effect of lowering the pKa of the external OH group, thus deprotonating it and enhancing its nucleophilicity. And so now the phosphate group of ACP is readily positioned for nucleophilic attack by this, forming ATP. This mechanism also explains why calcium and magnesium slightly increase the rate of the reaction, as we saw earlier, and these ions are able to displace the ferric ion from the ATP product and freeing the ferric ion so that it can go back to do another cycle of this. All in all, our results show that ACP can officially phosphorylate ADP to ATP, but only in the presence of ferric ions as catalyst. The reaction takes place in water and can occur in a wide range of aqueous environments. No other phosphorylating agent tested was as effective as ACP, and adenine is unique among canonical nucleobases in facilitating the phosphorylation of its nucleotide diphosphate to the triphosphate. So taken together, these findings suggest that the preeminence of ATP in biology has its roots in aqueous prebiotic chemistry. I would like first of all to thank my co-authors Cecilia Kuntz, Stuart Harrison, Sean Jordan, John Ward, Finn Werner, and Nick Lane, and the members of the Lane Origins group for their support during this study. Lastly, thank you very much for your attention.